in the water, the fire. I'll go wherever you go, in the valley, in the canyons. I'll go wherever you go. Hey lover, I'm ready now. Can't you see this right inside?
Well, good morning, everyone. I'm very happy to see so many of you over here, and thank you so much, Javier, for this nice historical touch, and Vero for a nice introduction. Thank you both for inviting me here, and also I owe you in-person apology for canceling my presentation in November. Due to, as Javier said, some health-related matters that are right now here with me, and they are 3 months old. Let's begin with the presentation. So today I would like to talk to you about the research interest of my group. We are very much interested in understanding how plants are exploring environments through the sensory receptors, and we are mostly devoted to understand what is the role of the cysteine-rich receptor like kinases. But let us start quite light. So as we all know, we as a human can be exposed to many different challenging instances. For instance, when we are too hot or too cold, we are mobile, so we can put some hat on or hide under the umbrella when it's too hot. However, what about plants? Plants are fixed in a space, so essentially they cannot run away in order to avoid all these environmental challenges. So they have to act locally, they have to perceive all the environmental stresses that they are challenged with a high precision, and they also need to respond to those stresses with the same precision. So how it is happening? So actually plants are very special when it comes to coping with stress. As I already said before, they are using specific sensory proteins, and those proteins are called receptor kinases. And when it comes to receptor kinases, plants are quite special because, on the contrary to other organisms, plants contain quite many receptor kinases, over 400 if we consider only Arabidopsis thaliana. And plants are not only special when it comes to the number of receptor kinases, but also when it comes to the diversity of these sensory proteins. So when it comes to receptor kinases, they are having usually this modular structure with the extracellular domain, transmembrane domain, as well as the kinase domain. And the diversity comes from the kinase domain, because many different families, currently we speak about 14 families of the receptor kinases, they display quite big diversity when it comes to the structure or architecture of the extracellular domain. And this also means that we have a quite big functional diversification when it comes to the what those receptor kinases are doing and why plants they have so many of them. And in the spotlight of today, we will have the family of the cysteine-rich receptor like kinases. But maybe before we will try to understand what's the role of this family, we can discuss briefly what's the canonical cascade of the signal perception and signal transduction in plants. So essentially, canonically, receptor kinases, for instance, the sensory receptors, they are sensing different signals outside of the cell, and then they transmit the signal to the inside of the cell. How they are doing so? So these sensory proteins, they canonically perceive certain types of the molecules, ligands, and they can be of a different variety, like peptides, glycans, hormones, you name it. And perception of this sensory, of this sensing molecules triggers interaction with other receptor kinases. And then the kinases, they are undergoing quite many different auto and transphosphorylation events. And this, in the consequence, triggers specific cellular responses. What is very frequently happening when these receptor proteins are activated, we are observing a massive production of reactive oxygen species in the extracellular space. And I will ask you to remember the presence of the reactive oxygen species because they will be important for what the CRKs are doing. Okay, so we know how the receptor kinases, they work canonically. However, what do we know about CRKs so far? Sadly, or maybe not sadly for our group, not so much. So we know that CRKs, this is the family of 44 members. They are divided into 5 phylogenetic groups based on the alignment of the extracellular domain amino acid sequence. And when it comes to the structure of the extracellular domain of the CRKs, it's quite similar between most of the members of the CRK family. And usually the extracellular domain consists of DAF26A domain and DAF26B domains. So they are kind of sitting one on the top of the other. And what is actually the biggest hallmark of this family of the proteins is the presence of the cysteine residues. So depending on the CRK, each member can contain from 10 up to 13 cysteine residues. And this is actually something what is very intriguing to me and to our group because, of course, cysteine residues, they can be quite interesting from the fact that very often in many different proteins, and I will tell you about that in a second, they can undergo in specific conditions certain modifications. We are talking here about oxidative modifications of the cysteines. Most of the cysteines, they have 3 thiol groups. And in the presence of reactive oxygen species, these 3 thiol groups, they can be oxidized into reversible and irreversible oxidative modifications. And most frequently, these reversible modifications, they alter protein structure and also the function. So essentially, this oxidation or the reduction processes that can occur on the cysteine residue, they can have a detrimental impact on the protein function. Okay, so I told you already a little bit about the CRKs and the fact that cysteines, they can be oxidized. Of course, later on, I will tell you what we found out about this. But probably you are also wondering, okay, in which instances these receptor kinases, they are exposed to the presence of the reactive oxygen species. And essentially, the answer is all the time. So throughout the entire development of plant, the reactive oxygen species, they play a very important role. At the beginning of the development and more frequently closer to the end, and here I'm speaking mostly about the transition from the vegetative to the generative phase of the development, but also throughout the entire life cycle, plants are exposed to many different environmental stresses, to many different pathogens, which are also triggering most frequently massive production of the reactive oxygen species. So actually, plants are coping with frost all the time. Right now, I just would like to give you a very short overview what are the research topics in our group and what we are the most interested in. Sadly, due to the time constraints, otherwise I will have to keep you here for 4 hours. I will talk about some chosen examples of what we are doing. So As I told you, we have a hypothesis that cysteines can get oxidatively modified in the extracellular domain of the receptor kinases. So we are using quite many proteome-wide mapping approaches in order to actually identify those modifications on the certain CRKs on a specific cysteine residues using redox mass spectrometry. Also, this oxidative modifications are assumed to alter protein function, which means we are also very much interested how the CRKs as the family interacts with each other, but also with other members of receptor kinases, and whether reactive oxygen species or redox regulation plays a role over here. We are also very much interested in understanding what is exactly the role of these cysteines, because CRKs are quite densely stuffed with many different cysteines, and some of them are predicted more to have a structural role. Some others, they are more of a functional cysteines, which are having this redox switching property. So that's also something which we are trying to understand, whether there is any certain categorization when it comes to different cysteines located in different parts of the extracellular domain. We also try to understand what is exactly the mechanism of the complex formation, so how it is happening, by what it is triggered, what is the interaction interface between two interacting partners, and actually how we can enhance, how we can modulate these interactions if we can. And the last part, which we started not long ago, I also told you that canonically, most of the receptor kinases that we know so far, they are activated because they perceive a ligand, a molecule. So we are also interested whether CRKs, apart from the fact that they can be modified by the reactive oxygen species, they also have an ability to perceive any other type of the molecule, and here we are quite focused on the glycans. Good. So, redox regulation of different proteins. So this is nothing new. In the field of the plant biology. There were in the past many different proteins that were identified to be redox regulated. For instance, BZR1, so the transcription factor of the brasenosteroid signaling. We know that this transcription factor in the presence of hydrogen peroxide gets oxidized on the certain cysteine, and this oxidation prompts its relocalization from the cytoplasm into the nucleus, and then interaction with certain other transcription factors in order to enhance BR responses. And also another example of this time receptor kinase. So HPCA1, that protein was published to be extracellular hydrogen peroxide sensor several years ago. So this is the canonical receptor kinase, and it was shown that extracellular domain contains 4 cysteines. And in the certain instances where there is a higher H2O2 or hydrogen peroxide content, these cysteines, they are forming disulfide bridges, and the formation of these disulfide bridges changes the structure of the extracellular domain. And this minor rearrangement of the structure can have downstream consequences. So, as I said, the concept is known, and we are right now wondering whether also it extends for the family of the cysteine-rich receptor-like kinases. So this is quite complex figure, I realize this, but I'm showing this for one reason, just to demonstrate to you how many different modifications on only one amino acid residues we can actually deal with. As I told you at the beginning, the cysteines or the free thiol groups, they can undergo either range or reversible modifications from the formation of the sulfanic acid disulfide bonds, newly discovered nose bridges or even double nose bridges, to irreversible modifications, which are most of the time actually not the modifications that can perform or give to the protein any specific function, but it's usually for the protein that is modified with these two types of the modification, essentially game over and the protein is being degraded. So we are mostly interested in understanding whether those modifications can occur on the extracellular domains of our cysteine-rich receptor-like kinases, and what's the function of these modifications. Let me pop all of this. It took us quite some years to understand which method will be best to map oxidative modifications on the cysteine residues. And to be honest, we are still using quite different combinations of different methods. So it's not like one suits for all. It really depends whether you are working with the proteins that are expressed in the in vitro system or you are working with the proteins in the in vivo arrangement. So what I will be talking to you about, these are proteins that we, or I should be more specific, extracellular domains that we expressed in insect cells. We purified those extracellular domains for all the members of the CRK family, so for 44 proteins, and then we perform so-called differential alkylation approach followed by mass spectrometry to map the presence of oxidation on the cysteine residues. So not going too much into these technical details because I think this we can leave out. So essentially what we were doing, we were having two types of the samples, so proteins, either non-treated or treated with certain reactive oxygen species, most of the time with the hydrogen peroxide. And of course, on every extracellular domain of the CRK, we had to consider that we might have a different arrangement or different ratio of different modifications, and also the oxidation we were expecting to be enhanced in the samples that were treated with the hydrogen peroxide. And so we were using differential labeling that allowed us to actually assess how much of the free thiol group we had before the treatment and how much was left after the treatment. So we were using the compound which is called acrylamide, and this acrylamide has the ability to label free thiol groups. So we are assuming that in the ROS-treated samples, we will have less thiol groups. So meaning lower amount of this acrylamide. So once we blocked already these free thiol groups, we were using the agent that reduced all other types of the modifications, and then we were using another type of the label called iodoacetamide to label newly established free thiol groups. And afterwards, we were performing just a typical MS sample preparation steps, and we were running our samples through mass spec. I have to say here, just for the simplicity, I use the trypsin for the digestion, but sadly, CRKs are very tricky, and sadly, CRKs are really glycosylated. So we had to use a combination of many different endopeptidases, as well as we had to get rid of the glycosylation in order to actually map the peptides that contain the cysteines. So this step is very simplified, but in fact it took us quite some time to understand what kind of modifications we have to apply over here. So all in all, we were able to identify, I think, 13 different CRKs that were differently labeled, so that they had a different ratio between the free thiol groups and the cysteines that were having certain oxidative modifications already in the non-treated samples, and they are listed over here. So those that were differently labeled, these are these bars that are representing the number of the cysteines. And also, this is just a very simple domain representation of the DAF26A and DAF26B. And if you can draw any very, very vague and basic conclusion, you would say probably that most of the oxidatively modified cysteines are actually happening on the DAF26A domain. And we also checked the availability of the cysteine residues in both domains, and indeed it seems that those residues that were more frequently labeled with different tags are more available when you look at the folded protein structure. Okay, so over here I'm just presenting a couple of examples of a few different CRKs for which we could very clearly see oxidation upon treatment with hydrogen peroxide. So essentially, if you expect the certain residue to be oxidatively modified, you would expect that in this bar you will have a lower ratio of the free thiol groups to other type of the modifications. So as you can see over here for this couple of examples of the CRKs, yes, we do observe a change, but this change is very minimal. So essentially there is just a small fraction of the cysteines that are being oxidatively modified in the pool of our proteins or a pool of our molecules. Whether that's enough to trigger certain biological responses, yes or not, we will see further. My time is over? No. Not yet, okay. Good. So the conclusion is that certain CRKs, indeed, they can be oxidatively modified on a certain cysteine residues. So right now we wanted to understand whether the presence of reactive oxygen species or the change redox conditions, they mean something for the protein-protein interaction. So we wanted to study whether ROS modulates the way how CRKs interact with each other. So again, we supported ourselves with the system that we already know very well, so the production of extracellular domains in insect cells. We produced two types of the proteins, bait that was fused with FC fragment, and FC fragment, as you know, has a quite high affinity to the protein A, and the prey proteins that were fused with the alkaline phosphatase. Alkaline phosphatase is an enzyme. If you add the substrate for alkaline phosphatase, then this will be colorimetric reaction, and the solution will turn blue, as is shown over here. So this is a very simple assay. You could compare it to the ELISA system. I guess you are all very familiar with what ELISA is. So what we were doing, we took, again, all 44 CRKs. We expressed them as bait, as the prey, and we fused baits on the bottom of this 96 protein A-coated plate, and we tested interactions between different baits against different preys, and then we measured the absorbance based on the colorimetric reaction with the alkaline phosphatase. And that, as you can see over here, it's really easy system, because apart from the fact that you can monitor plus minus, so there is interaction, there is no interaction, also based on the level of the absorbance, you can assess the strength of the interactions. So we did that for the CRK families. So we perform all by all interaction screen in, non-ROS and ROS conditions. And I will just right now walk you through what you are actually looking at. So these are reciprocal interactions that passed our stringent cutoff. So we just took interactions that were above certain value, and they occurred in both directions in non-ROS and ROS conditions. And we supported ourselves with the network studies in collaboration with Adam Mott lab at Toronto University. And we built these networks of interactions. So every circle here represents CRK extracellular domain. Those that are also surrounded by these white halos means that they are homodimerizing. And The size of the circle corresponds to the strength of interactions as well as number of interactions. And these different colors, these are different yeah, there is a lot of debate how to call them. I like to call them subnetwork. Some other people like to call them communities. But however, these zones of the closely interacting proteins in both conditions. And so as you can see, just like from the very brief view, the structure of these networks is quite different between these two different conditions. And certain interacting partners, they are also different per CRK. So for instance, let us take an example of CRK 18 over here. CRK 18 is a quite important node over here. It does homodimerize in the absence of reactive oxygen species. However, let me take a look because I'm a blind woman. Where is this? Where are you? No, yes, it's over here. Yes, indeed. Sorry, I should be better prepared with my own data. So, as you can see over here, CRK 2018 is indeed a hub of this subnetwork, and it dimerizes with itself quite strongly as well. You don't see the number here, but you have to believe me. But over here, in the ROS conditions, actually CRK 18 loses most of the interacting partners, and also it's not able to interact anymore with itself. And this is another, actually this is the most favorite example for me. This is CRK 28, probably you cannot see the label, you have to believe me this time. CRK 28 actually doesn't interact with anything else in the absence of reactive oxygen species. However, when we apply reactive oxygen species, CRK 28, let me just see. It's not very clear. Yeah. Yes, over here, exactly. Oh, I should put my glasses on, sorry. I wanted to look better, but the outcome maybe it's not better. Yes, exactly. Over here, CRK 28. So first of all, again, you don't see the numbers, but it homodimerizes way stronger with itself and also gains very strong interacting hub CRK 17. So essentially, the short story is that ROS rewires networks of interactions. And actually, this is quite complex to look at, and if you don't see very well, it's even more complex to look at. So what we decided to do, we decided to filter these interactions based on the expression of different CRK members at the different developmental stages, just to make sure whether they have actually, in planta, they have a chance to meet, whether they are expressed in the same developmental stages, in the same tissue type, so far, so on. So we decided to take two different developmental stages from the opposite sides of the plant life cycle, so the seedlings and the plants that are already entering the senescence. And essentially what you can observe here, that actually for the seedlings development or seedlings establishment, CRKs, they aren't that important. There are just few of them that are co-occurring with each other based on the transcriptional data, and that they form interactions. However, this is a completely different for the plants that are undergoing senescence. There are quite many members that can interact with each other and form these subnetworks. And also, depending on the level of ROS, these subnetworks, they can look completely different. So that was also something very interesting for us. I don't show it here, but it's known based on the RNA-seq data that we have that most of the CRKs are expressed in the senescence types of the tissues. And as you know, senescence is very much connected with production of reactive oxygen species. So that means that they are produced, they are expressed where the ROS production is also high. So, of course, it's nice to have this, but functionally, we wanted to focus on a couple of examples. And the most interesting for us for now, this is the homodimerization between 28 and homodimerization between 18. If you look at this graph, these two types of the proteins that are also belonging to different phylogenetic clades, they are behaving completely opposite in the presence of ROS, because the interaction between 28 is enhanced in the presence of ROS, and interaction or homodimerization between 18, it's depleted in the presence of ROS. So I won't have time to talk to you about this type of the interaction, which is mechanistically completely different from this one. And today I will focus mostly on the 28 homodimerization. Let me just take a sip. CRK28 dimerization. So at first we wanted to validate our results and check whether CRK28 indeed dimerizes or forms higher order oligomers. So for that we expressed our proteins in insect cells, but this time we used a large-scale expression using baculovirus stock. So we performed purification with the HISTAC and then size exclusion. So we were working with a quite nice and purified extracellular domain. And what you are looking at here, this is the Blue Native page. And these are different concentrations of CRK28 in non-ROS and ROS conditions. And as you can see for both, what was quite surprising to us, CRK28, of course, is present as a monomer, but also as a dimer. So this is something what we are observing with our in vitro screen, but also forms higher order oligomers. And it's really tricky to actually assess whether these higher order oligomers or even dimers are somewhat stronger in the ROS conditions. As I said, in both we are observing formation of dimers or higher order oligomers. Of course, this can be triggered by the purification conditions because we are purifying with HISTAC. HISTAC also can introduce certain types of the oxidation already on the proteins. So the question would be whether this is a consequence of the oxidative modifications that we were observing before. So that's something still what we are testing. Currently, knowing how to purify the extracellular domains of the CRKs, we are testing different binding assays and the thermal stabilities, also using different cysteine mutants. Some data are already here, so I will share those with you. But another aspect is, okay, we checked that with yet another method, CRK28 homodimerizes, but of course what is the most important is to validate whether this dimerization also occurs in planta. So it was published before by the group of Gita Cocker that CRK28 can homodimerize in Nicotiana bentamiana, and it's independent on the FLG22 treatment. They were trying to understand the role of 28 in defense responses, but for us the most important was that 28 is indeed, it does homodimerize in vivo. We performed a Fred film experiment where we tested interactions between CRK 28 in Nicotiana bentamiana, and again we could see and observe the interaction also on the level of the full-length receptor, not only on the level of the extracellular domain, which is actually a very good indication to start further biological investigation. Okay, but maybe coming back and sidetracking a little bit the dimerization, we were also very much interested what's the role of all cysteines, or at least those that we saw being oxidatively modified, on the stability of the full-length protein of the CRK28. So what you are looking at here, this is the transient expression in Nicotiana bentamiana. This is wild type 28, and it's nicely localizing to the plasma membrane, because we are also observing, these Hectian strands after plasmolysis. So this is actually what we would like to see. And if we are introducing certain point mutations on the cysteines that are forming disulfide bonds, either in DAF26A or DAF26B, we see that actually this expression pattern isn't really great. So the protein is not nicely localized to the plasma membrane. The expression is very patchy. That's also something that was kind of expected by us. However, here I'm showing just two examples of the two different point mutations, or types of the point mutations, but we tested all of them. And actually we found out that only mutations on the cysteines that are occurring in the DAF26B, one next to the other, doesn't cause this slight mislocalization of the protein. So essentially these 2 point mutations allow proteins still to localize very properly to the plasma membrane. On the top of that, we also mapped that actually these cysteines that are occur one next to the other, they are also getting oxidatively modified in vitro in the presence of reactive oxygen species. Good. So at the same time, of course, we knew already that we are going to follow on the biological or functional validation of what's the role of CRK28. So we ordered some transgenic lines, the knockout line and the overexpression line under the native promoter, and we started to look whether we can see any phenotypes. So on the level of the seedlings, because also CRK28 is very mildly expressed in the seedlings, we do not see actually any changes in the root length for none of these two transgenic lines. So they were not really distinguishable from Columbia plants. But when you start to track the development of these transgenic lines across the plant life cycle, actually at the stage of 4, most frequently 5-week-old plant, we started to see quite severe phenotype on the overexpression line and mild phenotype on the knockout line. So essentially what you are looking at here, these are 5-week-old rosettes of the wild type and overexpression line and the knockout line. And as you can see, the overexpression line is really, really not doing great. It's very small, is very chlorotic, and also we noticed that there is a disruption between transition from juvenile to adult leaves between these two different transgenic lines. So for overexpression line, it occurs too fast in comparison to Columbia, and for knockout line, too slow in comparison to Columbia. So essentially, on the level of the grown rosettes, we could see that CRK28 is very important for the proper rosette development and probably senescence. It seems that it does accelerate the senescence. And exactly that's what I have said over here, so I will not repeat anymore. So we performed quite a few assays in order to indeed quantify whether the senescence in these transgenic lines is somewhat disturbed. We performed very simple deattachment assay when we looked at the progression of the senescence on the deattached leaves of Arabidopsis tholiana of different transgenic lines. And as you can see over here, for the overexpression lines, that is abbreviated over here like 28-2, the progression of the senescence is way faster than for wild type or the knockout line. This was quantified based on the size of this chlorotic parts, and as well the chlorophyll content is also way lower in the overexpression line. So all those measured values, they tell us that indeed the senescence is progressing faster whenever we have a higher dosage of CRK28. But also, the phenotype that I have showed you for the 5-week-old rosette also corresponds very well with the autoimmunity phenotypes that are observed in the field for different proteins. So we have checked the ion leakage as a proxy for the programmed cell death, and in our overexpression line, the ion leakage is way higher than 2 other lines that we use for the control. And also, we know that the defense responses are way more enhanced in the overexpression line in comparison to Columbia or knockout line. So what we are looking at here, at the interplay between the senescence or autoimmunity signaling pathways that we are still trying to understand a little bit better, and how we did that. So for that, we supported ourselves with a quite extensive proteomics and phosphoproteomics experiment that we conducted. So we've taken this 5-week-old plants for the overexpression line as well as knockout line, and we tested what is happening on the level of the total proteome or the plasma membrane proteome, as well as total phosphoproteome and the plasma membrane phosphoproteome. And as expected, we found out that there are quite many different signaling pathways that are being disturbed in these different types of the plants. And this is briefly summarized over here. So essentially, most of the proteins that are accumulated in the overexpression line or phosphorylated in the overexpression lines, they are hallmark of senescence, a stress response, defense response, cell surface signaling, and programmed cell death. However, not much is happening for the plants that are actually lacking CRK28. Because what we observe that quite many different other CRKs also belonging to the same phylogenetic clade are overexpressed in the knockout line. So probably we have there some compensatory mechanisms that are trying to kick in, and some proteins that are connected with transport and cell surface signaling, but they were barely significant. So essentially, the lack of CRK28 does not do much. How am I doing on time? Okay, perfect. I will try to speak faster. Good. So, but what was very, very interesting to us was actually the results of the phosphoproteomics screen, because we could see very nicely that the additional dosage of CRK28 leads to phosphorylation on the CRK28 kinase domain, along with quite many hallmarks of the defense responses, like for instance phosphorylation of SOBIR, LIG2, BSK8, which is actually at the interface between plant growth and development. But also what was very important to us was the fact that actually the excess of CRK28 and phosphorylation on the kinase domain of CRK28 leads to phosphorylation of RBOHD. So we can probably conclude that the CRK28 promotes RBOHD for the stronger, more extensive ROS production, because all those 1, 2, 3, 4 serine residues were way more phosphorylated in the RBOHD protein. Okay, so we already with this proteomics/phosphoproteomics approach could see what are the signaling processes that are disturbed or altered in different CRK due to the different CRK28 gene dosage. But also we wanted to map what are the true physical interacting partners of the CRK28. And here we performed affinity purification experiment, and we decided to see actually what are the interacting partners through the entire development of CRK28 overexpression line? Simply due to the fact that we know that throughout the development, we have, first of all, increased senescence that comes along with an increased level of reactive oxygen species. So essentially, we performed affinity purification from 2 weeks old plants, 4 weeks old plants that are still before transition to the reproduction phase, and 5-week-old plant. And what we found out based on this experiment, that actually most of the proteins that are interacting with CRK28 at the stage of the 2-week-old plant, these are proteins that are connected with transport, with water transport, but also we have quite some plasma membrane ATPases. We have quite many aquaporins and also some proteins that are involved in the cell wall remodeling. So these were the interactors of CRK28 at the stage of 2 weeks old, when probably still the ROS levels are kind of well maintained. But this is something what is starting to change at the stage of 4-week-old rosettes, because what we are looking at here, these are actually quite many glutathione transferases that are interacting with CRK28. So it seems that the excess of CRK28 enhances ROS production, but also there are quite some compensatory mechanisms that are trying to deplete this to enhance ROS production. And I think the most interesting interacting partners we found for the 5-week-old rosettes, where we found quite many senescence markers, like for instance PR5. We found quite many proteins that belong to the defense response pathways, and quite many proteins that are connected with the transport, with the formation of the vesicles. Definitely we can say that across of the development, CRK28 interacts with many different interacting partners, and depending on the type of the interaction, different signaling pathways can be at that moment, at that developmental growth stage regulated. So essentially, this is exactly what I just have told you, that depending on the developmental stage, CRK28 interacts with different types of the proteins, first of all to maintain proper transport processes, then the redox homeostasis, and later on cope with all the proteins that are involved in the senescence or defense responses. The exact mechanism of why it is happening as such, we are still investigating, mostly by the involvement of the transgenic lines with the CRK point mutations. And do I have 5 minutes, or I should? Yeah? Okay. So lastly, what I wanted to talk to you about, because this is something what we are just beginning to work with, and I know that there are quite many people in here that are working with plant pathogen interactions. So that's something what I really would like your input on. So as I said before, most of the signaling, most of the sensing receptors, they rely on the interaction with different ligands, and we are also looking for the ligands for the CRKs. So it's completely unknown whether they can perceive any ligands, yes or not. So what we did, we undertook a little bit of the computational approach. So we tried to compute whether CRKs, they have an ability to bind something. So we performed so-called de novo modeling, where we were trying to see whether CRKs have ability to bind different types of the molecules. And essentially, we tested all the CRKs, which is not shown here, but all the CRKs as the single units, but also CRKs that can form the dimers. And based on this research, we could see that actually there are 3 different binding sites that can occur on the CRK extracellular domains. So that was already something that at least hypothetically, CRKs then can bind something. But also in the literature, what we know that that CRKs, they consist of the two DAF26 domains. And single DAF26 domain was shown in ginkgo biloba to bind mannose and give to the ginkgo biloba antifungal properties. And we checked whether the binding sites of the mannose of this single DAF26 domain also occurs in our CRKs. And we also find out that actually few members of the CRKs, they have very conserved binding motif of this single DAF26 domain. So what we did, we contacted the group of Fabian at the Boku University in Vienna. So that group is very specialized with a glycan array. So they produced for us the glycan array that is consisting of the cell wall components as well as the microbial glycans. And we selected a couple of CRKs that we wanted to test with this glycan array. And actually, we found one or probably more positive hits. And one of the CRKs, CRK41, that also has these conserved residues that are binding, that's supposed to bind mannose for the single GNK DAF26 domain, they also bind the glucan. When we mutate these residues, this binding does not occur anymore. So it's quite a promising avenue that we are currently testing. So we searched the literature and we found different types of the glycans that are derived either from the cell wall or they are produced by different microbes. So we enlarge even more the glycan array and we are trying to test all the remaining CRKs that we haven't tested yet to see whether some other type of the glycans they can bind to other members of the CRK family. And I think with that, probably I will already finish, not to keep you standing, Javier, for too long. So I think a very important part of the presentation is our acknowledgement. So, of course, thank you so much to my beautiful group. Probably that guy some of you can recognize already. So this is Sergio. Sergio graduated in November last year, and most of the work I talked about today, that's the content of his PhD project. But also many others, Chef and Viren, that were doing all the phosphoproteomics and redox proteomics results. Jente, she's interested in the structural properties of the CRKs. Judith follows on the glycan project, and Ran tries to understand interplay between CRKs and LRRKs. All our collaborators without whom we won't be able to do as much as we could. And of course, the money givers, because that's also a very important part. And thank you so much for your attention, and slide overdue.
Thank you very much, Elwira, for such a fantastic talk. Very, very, very interesting, very inspiring. And now it's time for you, for questions, just a little bit.
Hi, thanks Elwira for the talk, it was great.
Thank you.
I have maybe two questions. So I made one and then Miguel and then Mario again. So I was a little bit confused on the part where you were showing the phenotypes, because you said that basically it was the native promoter, but then you all the time says overexpressor. So it's an overexpressor or it's a native promoter?
Both. Actually, so it's a native overexpression in the background of the knockout line. However, with the qPCR experiment we performed, we know that we observe like almost 20 times overexpression in comparison to the native expression of CRK28 in an equivalent Columbia plant.
Okay, yeah, and in that case, can you exclude, because you know most of the time when you have like overexpressing line, you end up having like trans-silencing of like other members of the same family, especially in this kind of families that have many members. So did you check if all the other kinases are okay, or if they are silencing?
Well, I will not be lying to you by saying that we have checked all of them. We checked the closest homologs of CRK28, whether they are somehow disrupted by the overexpression of CRK28, and they aren't.
Okay.
So it seems that, yeah, this overexpression, which is still not very strong, doesn't actually have any influence on the expression level of all the at least closest members of the, or closest homologs of CRK28.
Okay, that's great. And the second question. So the redox proteomic is beautiful, but you see like this very small amount of like
Yeah.
oxidated protein. So I'm wondering whether this is just a matter of like a cell type. Have you considered to do single cell proteomics on those and see if you have like special cells with that? And it also it's important because you find the partners by in vitro systems. So are the partners expressed in the same cells or something like that?
Yeah, I think this is a really beautiful suggestion that I also had in my mind for quite a long time. But also, again, I have to be very honest with you that, you know, mapping oxidative modifications in the in vitro system is by far less difficult than trying to capture such modifications in vivo. So this is actually what we are struggling with at the moment, because we also realize that the redox proteomics approach that we have used for the in vitro expressed proteins, we cannot translate directly to mapping in vivo oxidative modifications. So right now we are upgrading our method. We have first quite promising results, but I think we are still very far from being confident to say whether what we see in vitro translates directly to what we can map in vivo. So yes, this is a beautiful approach, beautiful proxy, but not very easy to be used in the in vivo system. Thank you for your questions.
Yeah, great talk. I really enjoy a lot. I have kind of similar question about this line that you see a lot of phenotypes. So is this depending of the kinase activity? I mean,
Yeah.
so if you make a phosphodieet, then you wouldn't have that phenotype if you overexpress this, because you know then mechanistically it's going to be kind of different things. Maybe
Yeah.
you are just collapsing many of those other receptors, and the phenotype is caused because you have an increase of interactions, or is you actually need. And the other
Yeah.
question, then just do at the same time is so. Basically, what you see also would be an increased ROS, you know, in this transgenic line. So what happens if you mutate, if you make a CRISPR in this line of the NADPH oxidase? Do you stop having these defects?
Yeah, thank you so much, Mikael, for these questions. And I will begin with the first one regarding the kinase activity. Based on our work, we know that most of the members of CRK family, along with CRK28, they have active kinase domains. So, for instance, when we have the kinase that mutant, we don't see that severe phenotypes. And also that translates well to work that we do with the transient expression in Nicotiana bentamiana. So essentially, whenever we are working with the kinase that mutant, we can have a nicely expressed protein that is localizing to where it's supposed to go. However, with the kinase active domain, we see lots of symptoms of cell death, so the tissue collapses, and also there is a quite severe degradation of the protein, much more extensive than in the kinase that domain. Yeah. And in relation to your second question, whether this severe phenotype of CRK28 overexpression line under the native promoter will also occur in the RBOHD knockout background. We haven't tested that, so I really don't know. Yeah.
So, fantastic talk. Thank you.
Thank you.
So I have two questions.
Yeah.
When you check for or heterodimerization of the receptor kinase, you always need that the receptor kinase will form homoheterodimerization to be active. I mean, one silicon receptor kinase cannot be active.
So your question is that we have assumption that the receptor kinases, they are having active kinase domains?
No,
Or-
my question is if you always need that two receptor kinase be forming a dimer to be active, or they can be active. one single receptor. One
Oh,
single
I see what you are saying. Well, it really depends on the interacting partners. So, for instance, for CRK28, as well as for CRK E18, we know that the aspect of forming homodimer is very important for all the downstream implications. But we also test at the moment the crosstalk between the CRKs and LRRs. And actually, in most cases in there, that's also the case that we need the two partners to come together to actually trigger certain cellular responses. Yeah.
Okay. Thank you. And my second question is, I mean, maybe just I don't understand why a mutation in the cysteine affects the localization of the protein.
Oh yeah, maybe that's something what I did not actually explain well. So the crystal structures of the CRKs aren't available, so we don't know. We made a couple of attempts to crystallize CRKs, but it's on the way. However, we have a knowledge based on the closest homologs of CRKs named PDLPs, so plasmodesmata localized proteins, that they have exactly, or not exactly actually, they have a very similar extracellular domains. And based on the homology modeling, we know that most of the cysteines that are occurred in a conserved motifs in a DAF26A or B, they are forming disulfide bridges. So most of the cysteines that are forming disulfide bridges, they are considered to be structural proteins, structural cysteines. So disruptions of those will lead to the protein misfolding, and if protein is misfolded, this will lead to improper protein localization. But what differs CRK from the PDLPs is actually presence of additional free cysteines. And those free cysteines, at least according to our research, they are serving as this Rhodox or ROS switches, because most frequently we see them being oxidatively modified. And those are not having any implication when mutated on the proper protein localization.
Okay, now I understand.
Yeah.
Thank you.
Rafa, hey.
Thank you, Elwira, for your talk. So it's probably a very naive question, but it's somehow regarding to that one. So your group of a specific cysteine has always the same modification or can have several modifications? And if it does, how that happen? How can they distinguish between different modifications?
No, that's I think very relevant question, and that's still something what we are kind of investigating further, because at least based on what we can say at the moment in regard to our phosphoproteomics approach, most of the modifications that we are able to map is the formation of the sulfenic acid, so SOH. And it's very tricky to capture any other type of the modifications because that requires to use specific probes. Those probes are available, but they are mostly developed for the cell culture, for the animal tissues. So we have tested a couple of them that were coming from the Kate Caroline lab, who is like a guru in the cysteine oxidation field. However, I must say we were not very successful in using them in plants. So far we know that it's only either the formation of the disulfide bonds, because then we can test with the partial reduction experiment, or the formation of the sulfenic acid and DOT.
Hi, beautiful.
Hello.
Hello.
Hey.
Beautiful talk and beautiful work.
Thank you.
My question, I have two questions also. The first one is, you said that these proteins are heavily glycosylated. Do you know what, which types of glycosylations and what do they do?
It's a very tricky question to answer, because most of the time we are working with the extracellular domains that are expressed in the insect cells. We are using insect cells, and we can use only insect cells for that particular reasons, that only insect cells can somewhat mimic the glycosylation pattern of what can occur in plants. And you know, it does depend per expression batch, because different sugar moieties, they can be added differently depending on the expression. So of course we know, just based on the in silico predictions, what are the glycosylation we can expect based on the amino acid sequence. And actually right now we teamed up with the group of Yanni Bola at Oxford University. So they're gonna perform for us this glycoproteome screen to see actually what type of the glycosylation we are dealing with. But as I said, this will slightly differ per batch of the expression when it comes to the in vitro expressed extracellular domains.
And do you think they could be involved in the recognition of glycans from the last slide you say?
Yeah.
That they could be like sugar.
So also what I actually did not share with you, but maybe that will be quite relevant as well for the function of the CRKs. Because with the SECMALs, so size exclusion connected with the function that allows us to estimate the mass of the protein along with the mass of the glycosylations, we know that the size of the glycosylations differs per CRK. So for instance, CRK18 I did not talk about. For CRK18, glycosylations add up 10 kilodaltons. For 28, it's only 3 kilodaltons. And yes, of course, it's nothing what we have tested so far, but I have an underskin feeling that this glycosylation will be also important feature for the glycan perception.
Thanks. Yeah. And the second question is short.
Yeah.
Have you tried to suppress the autoimmune phenotype, like knocking out key components of the immune signaling pathway?
No, not yet. This is still quite undeveloped part of the research. So these are all the things that we have in the pipeline. But as we all know, making all the transgenics takes quite a while, even with CRISPR-Cas.
Any other question? Just to finish, I would like to do a very short question. You have shown that some cysteine residues of CRK28 are very important for functionality, and furthermore, you have shown that the ectopic expression of CRK28 modifies the phenotype strongly. Have you checked, probably you have mentioned, but I don't remember, have you checked the effect of ectopic expression of mutated CRK28 in Arabidopsis, and also has a very strong effect?
The lines are under selection now.
Yeah.
So, I mean, from what I can say, but you know, take my word with a grain of salt, actually these point mutations, they do not lead to such a severe phenotype. But as I said, you know, I mean, we still don't deal with truly homozygous lines, so I would not, you know, say that is 100%, but it seems this way, that the phenotype is not that severe. Yeah,
and just a technical question, why did you substitute the cysteine by alanine and not by serine? Is there any special reason for that, or just simply because?
Well, yeah, so no, not really. I think that there are two different teams in the field of the cysteine work. So people who are substituting with serine, or people who are substituting with alanine. And of course, there are certain reasonings for one or the other. And we thought that, you know, substituting, I mean, because we are very much focused on the structural properties, that substituting with alanine, something what is way smaller, would probably has lesser implications on the protein structure per se than substituting with serine. So, I mean, we have chosen the alanine at the beginning when the project has started, And later on, just to stay consistent, we kept on working with the alanines. We also have, because, just to be able to handle questions as such, we also made a couple of mutants with the serine just to see whether actually different substitutions would mean anything for the protein expression or the localization. And those that are substituted with serine, they behave as those with alanine. So we can be kind of course, not 100%, but quite confident that it actually does not really matter.
Yeah, okay.
But I'm not married to one or the other too much, so.
Thank you very much for your nice
thank you so much.
answers, and always also congratulations.
Thank you.
Okay,
Yeah.
just I will.
In the water, the fire, I'll go wherever you go. In the valley, in the canyons, I'll go wherever you go. Hey lover, I'm ready now. Can't you see this right?